Okay, good morning and good afternoon, everyone. For the next 30 minutes or so, I will be discussing some advances in vaccine and immunization technologies. This is my conflict of interest statement. So I've been giving this presentation for the last several years at ADVAC, and every year, right before the presentation, I look back at the past year to update my presentation and see what has happened that was of interest that I should include in my presentation. And this year, my job was very, very easy because 2020 was truly an extraordinary year for vaccine technologies, obviously because of the COVID-19 pandemic. So I decided to use the COVID-19 pandemic as a way to structure my presentation and how the pandemic has pushed vaccine technologies forward, and more specifically, how we were able to develop vaccines at record speed and approve them thanks to rational vaccine design. We were able to prove that the new vaccine technology, mRNA, was safe and effective, and how we were able to expedite vaccine manufacturing at mass scale, which was an unprecedented effort. And also, I'll spend a few minutes to talk a little bit about efforts to develop thermostable vaccines that can be used worldwide. And then I'll end by giving you some thoughts of what I think is next. So traditionally, vaccines development has been an empirical process where vaccines were traditionally developed either by inactivating all viruses, for example, like the rabies vaccine or the Salk polio vaccine, or by developing attenuated viruses through animal passage or tissue culture passage, such as the measles and mumps rubella vaccine. However, more recently, we have been able to develop vaccines using a more rational approach by harnessing the knowledge of what constitutes an effective immune response to design a vaccine, and to harness this knowledge to develop a vaccine that elicits the desirable immune response. And this rational vaccine design has been enabled by development of new technologies, most importantly, technologies in immunology and also in structural biology. So how does rational vaccine design process works? So what we do is that we learn from an infected person or a person has been vaccinated. We isolate from the sera the immune cells. Then we identify from these immune sera what are the most strongly neutralizing antibodies by mapping them through structural biology onto the antigen. And then design a vaccine antigen that really concentrates the epitopes that target the strongly neutralizing antibodies. And we evaluate these vaccines in animal models. We measure the immune response in animal models to prove that, in fact, the immune responses are the ones that we wanted. And if we need to tweak the vaccine, we go back to the beginning of the process, we tweak the vaccine some more, and we do this in an iterative manner until we find the vaccine that we want. So, and let me give an example of how this has helped with the development of an RSV vaccine currently in late-stage clinical development. So structural biology studies have shown that the main antigenic target of the RSV virus, which is the protein F, comes in two different shapes. Can come either as a pre-fusion form, which is the one in the middle of the slide that looks like a ball, or it comes as a post-fusion form, which looks more like a tall and long shape. When the scientists went to map where the strongly neutralizing antibodies from people that got infected were binding onto these two different types of proteins, what they noticed here, so in the left, you can see that the strong neutralizing antibodies are indicated in red and orange. What they noticed is that the most strongly neutralizing antibodies were binding preferentially to the pre-fusion form of the protein on the left, and not so much on the post-fusion protein on the right. So the scientists decided then that to make a good vaccine for RSV, you need to lock the protein into the pre-fusion form, which is this ball, and this should enable the development of strongly neutralizing antibodies. So they actually adapted this knowledge to other viruses, such as coronaviruses in the last several years, where the mechanism of entry of coronavirus is similar to the mechanism of entry of paramyxoviruses, where RSV is the family of the RSV virus. So they decided to apply the same principle, and they found that even in coronaviruses, this is several years back before COVID-19, most of the neutralizing antibodies were binding to the pre-fusion form. So what they did, they in these yellow and orange squares there, in that portion of the genetic code for the F protein, they inserted two prolines in a way to stabilize the protein in the pre-fusion form in the ball. And so they've done trials to show that, in fact, when you have this kind of stabilized protein, you have a very effective immune response. So when COVID-19 happened in early last year, in 2020, our scientists at their NIH in the Vaccine Research Center, which had done this work before, they already knew how to design the antigen for another coronavirus, in this case was SARS-CoV-2. Now the question was, what platform do we use to this antigen? And they decided to try this new, relatively new technology, not completely new, but never been proven before in an efficacy trial, which was the mRNA vaccine platform. The reason why that they decided that very early on last year to try the mRNA platform was because it's very, very rapid to produce the vaccine. So you only need the genetic sequence of the pathogen to really quickly make a lot of the vaccine and then move into the clinic. It's safe and non-toxic. Since it's not just it's safe, it doesn't integrate, and the RNA degrades quickly in the body. Because the antigen is expressed intracellularly, you get a good immune response, both humoral and cellular. And also because you don't have an actual viral vector, you don't have the problem of anti-vector immunity. So you can give repeated immunizations over and over again if needed. The disadvantages, of course, that we knew was this technology had never been really proven before because we didn't have any licensed vaccines. Booster shots might be required, and in the beginning there were problems with stability, and some RNA vaccines require ultra cold storage. But all said, our scientists decided early on to work with an mRNA vaccine company, in that case was Moderna, to apply the knowledge of how to make an antigen by using this new mRNA technology. And so this is how you make the vaccine. You basically have the sequence. With a computer, you engineer the RNA sequence to insert these two proline mutations so that the protein gets stuck in the pre-fusion protein. You express this engineered RNA into a circular DNA plasmid, and you use the DNA plasmid to in vitro produce mRNA molecules that are then encapsulated into lipid nanoparticles. And this is what ends up in the vial that gets injected into the muscle. So this graphs, so this is the timeline of how quickly they were able to evaluate and get preliminary approval of the Moderna mRNA vaccine. So the sequence of the virus was released on January 10th. It took 5 days for NIH scientists to actually design the correct antigen and put it in GMP production. And it took 2 months from the sequence publication to the phase 1 trial. It took another 2 months to go into phase 2 trials, and another 2 months to start phase 3 trials. So this was really an unprecedented speed to really move from the sequence of a pathogen to initiating a phase 3 trial. And then, as you all know, as we received the first results of the trial of this vaccine phase 3 trials in November of 2020. But of course, we knew that, in the beginning, we didn't know if the mRNA was going to work. It was everything was moving quickly, and we knew we needed a variety of different approaches for vaccines to diversify our vaccine armamentarium and also to diversify the producers, because we knew that there was going to be an enormous need of vaccine worldwide. So another approach that was leveraged very early on was the use of adenovirus vector vaccines. And two companies used this approach, Janssen and Oxford and AstraZeneca. In this case, you use the adenovirus vector that doesn't replicate. You clone the protein of the SARS-CoV-2, and then this virus be delivered as a vaccine. And the way that you do it is also pretty simple. You clone the gene into the plasmid expressing this replication incompetent virus. Then you transfect these into cells that provide the replication proteins, because this virus doesn't replicate because it doesn't have a replication component. So you have the cells providing this protein that allows for the virus to replicate, and that's how you produce the vaccine. So the advantages of this platform, of course, is that this was a very well technology, unlike the mRNA. It's plug and play because you already have these vectors ready to go. You just have to insert the new genetic sequence of your target antigen. You have a very strong immune response involving both T cells and B cells. It's thermostable, which is, we know that it's thermostable for several months in the refrigerator, which is a huge advantage when you're trying to deliver this vaccine in very remote parts. And because it's very easy to manufacture, you can scale it up and it comes at low cost. So there were definitely some very strong advantages. The disadvantages was that for adenoviruses, we know that previous exposure to some adenoviruses can lower the effectiveness of the vaccine. And also it's relatively complex to manufacture. It's more complex than mRNA vaccine, and that's why they arrived into the market, let's say, a little after the mRNA vaccine. And then there was a third approach. There were several approaches. I'm just going to mention some of the ones that the US government really focused on for the most part. It was the protein subunit, and there were two companies that the US government supported, Novavax and Sanofi, in collaboration with GSK. And in this case, you basically have a purified protein, the spike protein from the virus, mixed with the GSK adjuvant and injected. The advantages of this is, of course, this is a very well-established technology. It's suitable for people with compromised immune systems. You don't have any living components, and is relatively stable. The disadvantage is that this is a little more complex to manufacture because now you have to purify the protein. You might need adjuvants and booster shots, and also it's a little. It can be a little challenging to figure out the right combination between antigen and adjuvant when you make these kind of vaccines. And then there were other hybrid approaches where other companies are developing these, what we call them as heterologous prime and boost, where they prime a person with one type of vaccine. In this case, this is the company Gridstone, is the chimpanzee adenovirus base vaccine with a different type of vaccine. In this case, is a self-amplifying mRNA vaccine. And so this approach is currently in phase 1 trials. Let me switch gear and talk a little bit about vaccine manufacturing. We realized in the last year how important it is to have a reliable, efficient, and low-cost manufacturing platforms that allow for on-demand production. So we had to ramp up production really quickly. So efficiency was really key in the past year. So there are 4 different ways that we can manufacture vaccines. Main ways, there are others as well, but through culture cells, through plants, and egg-based. And I'm going to give you some examples about the first, and tell you a little more. So the main steps in biomanufacturing are really 3. Most vaccines are grown in cell culture. So you have to first grow the pathogen or at least even the protein, express the protein. Then you have to purify your antigen. And there is a variety of ways to do this through concentration and chromatography. And then there is a quality control step where you need to ensure that there is sufficient purity, potency, stability, there's no aggregates. And once the vaccine has passed all these 3 steps, then you can deploy it. And there are recent innovations in biomanufacturing. The main 3 innovations are the use of single-use bioreactors. It used to be the bioreactors had to be cleaned up after every run and decontaminated and recertified. Now they use bioreactors with these single-use plastic bags. They can be disposed of, which allows for greater flexibility, lowering the risk of contamination, and a ready-to-use manufacturing room as needed. They're automating the way that you do the quality control steps. So you can monitor automatically throughout the manufacturing process all the critical quality and performance attributes of your vaccine. And also another recent advance that might become more mainstream in the future is continuous manufacturing versus batch manufacturing. So instead of having the separate 3 steps I just told you, where you express the protein, you purify, and then you do QC, you have all the steps attached as a continuous flow, which improves efficiency and control, allows for smaller footprint and reduce the cost. And I'll give an example of that. So the mRNA vaccines are the first cell-free system. It's the first time that we manufacture a vaccine that goes into people that has been made in a cell-free system way. So the advantage of having a cell-free system way to manufacture vaccines is that it's done in vitro. You don't have any impurities and contamination from the cells, so the purification process is a little bit easier. You just need the components that the mRNA that you need to do an in vitro transcription. It's very flexible and standardizable because you just change the sequence of your plasmid, and you can make a new mRNA. So the sequence doesn't affect the yield usually. This is how mRNA vaccines are made. You have the production step, as I told you earlier. In the mRNA vaccine, you can do a 2-step reaction where you first make the RNA and then you add the cap at the 5' end, which is important for translating, is important to allow the cell to recognize this mRNA as an endogenous mRNA and express it as an endogenous protein. Or you can do the same in a 1-step reaction. There are advantages and disadvantages to both. Then you have a purification step, as I said earlier, and depending on what scale you're making the mRNA, you can do, if it's lab scale or large manufacturing scale, you have different technologies, usually involving some sort of chromatography to purify the mRNA. And then you formulate the mRNA with the lipid nanoparticle, and then you do fill and finish, and you put in the 2 vials and you deliver it. The challenges of this manufacturing technology is the cost of reagents. So the reagents to manufacture mRNA in vitro are expensive. This is changing because of the demand has increased. In fact, and also the scale up. In the beginning, we were really worried about the ability to be able to scale up manufacturing, but we are making huge progress very rapidly, and we expect to manufacture 2.5 billion vaccines, mRNA vaccines, in 2021. And this is an example of the continuous manufacturing. So this is how they're envisioning they could in the future manufacture mRNA vaccines in a continuous flow, where you have production reaction, and then this is connected to a processing step where you purify the mRNA. The enzyme that wasn't used gets recycled, goes back to the production. And then if you continue in the flow, you have the capping reaction. Again, after the capping, it goes to a 2nd step of purification. The capping enzyme gets recycled. And then finally, you have the purified product. And so the advantage of this is by recycling components, you can have a less expensive product, and you can do it faster, and it's more less prone to errors. So for the adenovirus manufacturing, that's done quite differently. You need to grow these viruses. So because the adenoviruses are replication deficient, so they made a replication deficient to make them safe, to make them to be more safe. And to do that, they remove the E1 gene from the adenovirus. The virus by itself cannot replicate, the virus cannot spread to other cells, can express the antigen but cannot spread. So in order to grow the vaccine, you need a cell line that provides this E1 protein to allow the virus to replicate enough so that you have the vaccine. You don't need high biocontainment for adenovirus, which is an advantage. And so you need the cell lines, however, that stably express this E1, and they're adapted to grow serum-free and multiply in suspensions at very high density in bioreactors. So this vaccine is grown in cells in bioreactors, in the special cells that provide the E1 protein. And there is one of these cell lines is the PERC6 that Janssen has developed, and is a very efficient cell line as it allows, you can produce over 1 million doses of vaccines in a 1,000 liter bioreactor. And then I just want to say a word about the plant-based manufacturing. There are some companies that are developing SARS-CoV-2 vaccines using plants. This example is from Medicago in US, where you basically synthesize the gene of interest. You infiltrate the genetic material into plants through a vacuum system. Then the plants grow, then you harvest the plant, and then you purify the antigen. And the Medicago SARS-CoV-2 vaccine is currently in phase 3 trials. And this is what the VLP particles look like. A couple of words about vaccine delivery. So of course, most of the vaccines that we know are delivered through needle and syringe. We also last year realized how we need to diversify the way to deliver vaccines, because in the beginning there was a shortage of, we had to really crank up production of needles and syringes, because all of a sudden it was a very large need for these tools. So there's a lot of efforts to develop different technologies to deliver vaccines. I will just talk about a little bit about microneedles and then a little bit about nanoparticles. And if you look in nanoparticles with the liposome, this is similar to the way that we deliver the mRNA vaccine. We have a lipid nanoparticle that contains the RNA vaccine, and that's how the RNA vaccine gets delivered into the cell upon injection. So the microneedles are a relatively new technology where you can actually make so you have a little tiny patch. There is the picture in the middle here on the top. It's a tiny little patch of a few millimeters that has these microneedles containing the vaccine. It can be formulated in different ways. In this example, the microneedle dissolves into the skin, and as dissolves, it releases the antigen. And the interesting part about microneedles is that the microneedles are made of such length that they release the antigen into the dermis, which is where a lot of the Langerhans cells and other immune cells are present, which can capture the antigen and initiate a very strong immune response. You can inject also vaccines intradermally with needles, but it's very difficult. You have to get the right needle and you have to be very skilled at doing that. With a patch is quite a bit easier. And in this example on the right, this was a study done at University of Pittsburgh and at Carnegie Mellon, where they tested the HA antigen from influenza, and they compared the immune responses between microneedle, dissolvable microneedle, and hypodermic needle. And you can see that you can get a very effective immune response. I believe this was done in monkeys. With the similar amounts of vaccine, you get very strong response with the microneedle versus regular needle and syringe. Just a quick word about thermostability. There are a lot of efforts to figure out how to make the vaccines more stable at room temperature. This is really makes vaccination campaigns a lot easier and less logistically challenging, and also lowers the cost of the vaccine because you don't have to replenish the stored vaccine and waste. So there are two different types of vaccines. There are liquid vaccines on the left here, and these are easier to these are they're already diluted and ready to go, so they're easier to use and to administer, but they tend to be a little less thermostable. And then on the opposite side of the spectrum on the right is vaccines that are lyophilized, so they're more thermostable, but then it's a little more difficult to administer because now you have to dilute them, you introduce risk of contamination as you resuspend the vaccine, and so forth. So we're trying to figure out better ways to thermostabilize the vaccines there and keep them in a liquid form if possible. And so the thermostabilized vaccines are in a liquid form by using a variety of antioxidants and other excipients, by using gases that are on top of the vial, and also sometimes nanoparticles to stabilize this. And on the on the right part of the slide shows how they lyophilize vaccines, where they do different physical techniques like spray drying and alternative drying methods. So and sometimes also with excipients, so that when you resuspend the vaccine, it resuspends well, it doesn't make clumps, and it doesn't lose potency. So there is a lot of work currently going on, and the goal is to make vaccines, they're designed from the very beginning to be thermostable, so they're more easily used worldwide. So what is next? I think one of the technologies that will probably become more and more, you probably hear more and more about, is nanoparticles, but not regular nanoparticles like the lipid nanoparticles. These are engineered bioparticles, nanoparticles by researchers. So by using structural-based design, what our scientists are doing is they're making these backbones for nanoparticles. They're shown here in gray and yellow. And by mixing different components using some structural design, this core, the nanoparticle, enables the attachment of a different number of antigens on its surface. So in this case, you have this blue protein that is attached, and you get a vaccine that looks like that. And depending on what the core structure of this nanoparticle looks like, which you can change, you can make nanoparticle vaccines to look in a variety of different ways. So you can decide how many antigens, for example, decorate the surface of this nanoparticle, how big or how small they are, which might, so you have a lot of control over the type of immune response and the site where the vaccine reaches. So they're currently developing a SARS-CoV-2 nanoparticle-based vaccine. In this case, you have a vaccine that has 60 spike receptor binding domains on the vaccine. And this vaccine is currently in phase 1-2 clinical trials in US. And by the same technology, they're developing vaccines for influenza, hepatitis C, and RSV. And another thing I like to mention is the concept of precision vaccines. So we're learning that one size fits all doesn't work for a lot of people. As we know, the immune response to vaccines in the elderly or in infants is quite different from the immune responses in healthy adults. So we're trying to figure out how we can make vaccines that are more customized to the target population that will receive the vaccine. And a lot of, to do this, we are leveraging new omics technologies. In this picture, you see a baby receiving vaccine. Instead of measuring, say, the neutralizing antibody responses, or the binding antibody responses, which is the normal thing that we measure after a trial, if you employ metabolomics, proteomics, and transcriptomics, you can look at very fine differences in the immune response. You have a more of a holistic view about the immune response to vaccine, and you can then tweak the vaccine to induce more or less of an immune response that is desirable in a certain type of population. And once you have a vaccine, you tweak it, you test in the appropriate animal model, and then you put it back into a clinical trial to see if you have improved the vaccine. So finally, I'd like to leave you with some conclusions. So I guess the silver lining of this terrible pandemic that we're still living is that the pandemic has catalyzed a major renaissance in vaccine technologies. There was a huge amount of progress that was made in the last year, and we're continuing to make progress. And these new technologies not only are dealing with COVID, with SARS-CoV-2, but also we hope that will help advance other important vaccine areas such as HIV, tuberculosis, malaria, and influenza, where there's still important challenges that we need to solve. So hopefully we're going to learn from these, whatever we're learning in the last year is going to help other fields as well. And one word of caution, however, the vaccine technologies do not solve all the problems that we have. It's really of critical importance that we understand the basic biology of each pathogen, or at least of each family of pathogens, because that's really the first step to really design an effective vaccine. So in each family of pathogens, we need to figure out what is the correct antigen or epitope combination, what is the correct adjuvant, what is the correct conformation, what is the correct expression platform for each antigen. And with that, I am done, and thank you very much for the attention.